Good evening to all the participants. Welcome to the day 14 session on tools and servers for computational vaccine design. So we also discuss a case study, few case studies in vaccine design on today, and the final day from tomorrow onwards. Okay, we extend the MCQ test up to Saturday. That is, I'll be releasing you the overall feedback form and MCQ test form on today. That is after 8 p.m. Later, the deadline for completion of both the forms is extended up to 1st of August. Okay, so 4 days are provided to you, so you can able to complete it. Because why I have extended is you can learn if you have missed any videos, you can watch the recorded videos and you can try to complete without watching simply AI search tools. Okay, so watch the videos and you can answer accordingly. But the in MCQ I ask very basic things, okay, basic questions I am asking. This is just for self-assessment, not going to reflect in the certificate, whatever marks you will be getting. Okay, and related to today's session, before going to today's session, can we recap the last session topics? What exactly did we discuss on last session? We discussed something on B cell epitope prediction, isn't it? So give me examples of B cell prediction, B cell epitope prediction algorithms. So this is my first question. So we have seen so many B cell epitope prediction algorithms in the IEDB itself. Mention few. In my PowerPoint, I've shown more than 20 web servers for B cell epitope prediction. Can you mention few algorithms present in IEDB web server for B cell epitope? So if I ask you in MCQ, we are trying well in coding, but how about this kind of descriptive answers? Do you remember PepiPred, PepiPred 2.0, and yesterday we have practiced PepiPred 3.0, Elite Pro, Disk Pro, okay, Discotope spelling is wrong. Discotope. This is what we have seen majorly, isn't it? Other than this, surface accessibility, Polansker and Tonko antigenicity, Corpus and Skulls flexibility. Okay, so these are the other algorithms we have discussed, isn't it? Yes, very good. PepiPred, ABC Pred, good. So remember all this, okay? Minimum few tools and practice at least one or two tools so that you can get an idea of what exactly you're doing and try to bring in the form of an outcome, okay, by doing the research. Now let me take you to the day 14 session on tools and databases for computational vaccine design. Initially, we discussed about vaccine databases, then vaccine tools, and then the types of vaccines for prevention of diseases. So I'm not going to take much time on today. So I'll be completing as soon as possible, and I will be circulating you all the forms through mail and WhatsApp. You can try to answer from now onwards. I'm not going to limit to provide you the certificates until Saturday. Even if you can able to complete within 1 or 2 days, I can also give a special preference to send you as soon as possible to your email. Okay, so try to complete as soon as possible. Now let me take you about the first uh, in this slide. First, I am mentioning you about human vaccines. This is a HUVAX. This is for licensed human vaccines. Okay, you can able to get the information from violinet.org HUVAX. Here, what you can give as an input, select any pathogen. If you select any pathogen and simply search, so here you can find vaccines for most of the pathogens, okay, human-based pathogens in HUVAX. Only thing is that you have to use the select, and if you want to use advanced filters, you can use any of these. And VVACs, this is for licensed veterinary vaccines. Okay, you have to select any specific animal, poultry like hen and chicken and what else? Animal we consume, goat, cow. All these things are fine. You can able to find here. Even pet animals like dogs, cats, camels. These are also available here, and any specific pathogens. So this and this you have to select. So this parameter and this parameter. That's it. You have to simply click search. So these are the websites where you can able to see the vaccines already developed for humans and the animals. If the vaccines are not yet developed for few pathogens, then you can use the B cell epitope, T cell epitope, whatever we discussed on the previous 2 day sessions. Got it. So first we have to check what vaccines are available. So for this you can go to this website. The next thing is violinate protein. Okay, this is actually you can able to find the protective antigens. In the vaccine, we generally have antigens. To increase the efficacy of antigen, we use adjuvant, right? So antigens are very important. So if we want to find the protective antigens, okay, especially targeted by the acquired immune response of the host, then we have to use this. So for this, the first input you provide is the pathogen or disease name. Okay, so this itself is sufficient. If you search, you can find plenty of antigens available for this particular species as well as for the disease. Okay, like this you can search. At present, Protein has updated up to 1631 protective antigens. The next is DNA vaccines. Okay, for a DNA vaccine, we have a separate database, DNA Vax TB. Here you have plenty of tools for DNA for coming up with a DNA vaccine. Actually, DNA Vax query. So it takes generally a query DNA vaccines and which is experimentally verified and stored. So whatever experimentally determined, okay, that kind of sequences of the DNA vaccines are available here. And DNA Vax plasmids, specifically we have also for plasmids. DNA Vax antigen, specifically we have a separate link. And if you want to blast DNA or protein sequence against experimentally verified DNA vaccines, you can use this DNA Vax AG blast. So these are the major tools you can find in DNA vaccine database. Okay, this is especially can find for humans. And similarly, Vax vectors. This is a database of recombinant vaccine. Nowadays most of the recombinant vaccine vectors came into existence. Even for treatment of SARS-CoV-2, you can find much of recombinant vaccine vectors and researchers. So to come up with these kind of vaccines, you can use VaxVec vectors, VaxVec vaccines. You can find the numerous recombinant vector vaccines. Similarly, for antigens, we can find. Okay. Next, we'll be discussing on Virmogen TB. According, am I audible to everyone? Yes. Am I audible to everyone? Yes or no? Yeah, participants, am I audible or not?
Yes, sir.
Yeah, to maintain in silence. I think the last session you want to make silent. Okay, I'm audible. Okay, let me take you to the PowerPoint. I was explaining you something on uh, the virulent genes. In the live attenuated vaccines, you heard about the types of vaccines, live attenuated vaccines, killed vaccines. Okay, like generally, if you want to make viruses inactive, so how can we make viruses inactive? Either use heat treatment or just use a chemical treatment so that the virulent genes present in the virus is going to be in silent. Okay, the virulent genes won't be get activated, and so whatever virus, okay, with that virulent genes, whatever we made passive, okay, this can be used for prevention of upcoming of virulent genes possessing virus. Got it? We inactivate the viruses, that is, we do the gene silencing with the virulent genes through heat activation or through chemical activation. So for that we have Birmugen TB. So if you see, plenty of examples are provided. Again, here they have given first example as Mycobacterium tuberculosis. Check with this, you can find Birmugens. Vaxar, vaccine animal response. So what are the animals we can able to find, and corresponding vaccines we can find in this database Vaxar. We find plenty of pathogens, and you can see around 22 different animals are mentioned here. Okay, if it is mentioned as hyphen, it means that we don't have vaccines for human. Okay, so but we have for actinobacillus fluoropneumoniae, we have vaccines for mouse. How many? 10 vaccines we have for mice. Similarly, we have 5 vaccines for pig. Okay, it depends on types of pathogens. Vaccine and immune response. Now let me take you to the vaccine. If you want to find what are the vaccine mechanisms, we can go with this tool called vaccine. Okay, so we have to simply go to the introduction part to find what is Actinobacillus pneumoniae. Actually, this is a disease generally occurring in the porcine. Okay, and if you want to find about the pathogenesis information, types of animals you can check for the treatment or prevention of disease. Especially here, it is vaccine essential for prevention, protective immunity, and number of vaccines discovered at present is now 21 numbers. So like this, we have series of pathogens. Okay, we have plenty of pathogens, and for pathogens, we have the mechanisms are mentioned. Instead of going to the chat GPT, we can able to find the information from here. The next is something related to rational vaccinology. How we can use the procedure to design vaccines through VBRC nerve tool. This is one of the tool we can use for predicting vaccines. Okay, VBRC nerve tool. So initially, if you start from the step one, this is actually step one. We determine the immunogenicity and protective efficiency in small model like mice, rat, etc. Not human primate model, can be up to chimpanzees. So whatever we determine the immunogenicity and protective efficiency, okay, of the particular vaccine, okay, this is the step number one. For doing this, we can use these tools. So whatever tools that shown is related to the B cell and T cell epitope. Here these tools also has specific tools to determine immunogenicity, other than finding the B cell and T cell epitopes based on scores. Okay. The next step, after predicting the epitopes through any of these tools, the second step is to go with optimization of factors affecting codon usage. Eliminate stable mRNA admins. Yesterday also, one of the participants asked me like how to make a protein more stable. I told her better also check with any kind of beta folds are formed, any hairpins. If you see the mRNA, you can find similarly hairpins. Okay, you should not find the mRNA can have hairpins. If you find mRNA is having hairpins, then the stability is less actually. Eliminate nucleotide repeats. Eliminate possible allergenic sites. This is step number two. After predicting the epitopes. For doing this, you can use ATGPR codon, STAR, Signal B, Gene Composer, ALG. Few tools are actually, most of the tools are commercial tools. The third step, what we make is the determination of transcriptional unit for gene expression. So one should be able to find the transcriptional unit, okay, transcriptor genes. So these are all also important for a specific target, for specific pathogens. So whatever we go with the transcriptional unit, we can check from promoter scan, polyaprid. So these are the tools we use. Later, finally, from these tools in the sequence, if you use it in pipeline, finally we can come up with a potential vaccine candidate. This is the final vaccine we can able to come if you use these steps in pipeline. Now the next thing is computational strategies for the development of potent vaccine, including antigen processing. What are the different computational strategies? So from yesterday onwards, we are discussing much on epitope prediction tools. The epitope prediction tools stands under method one. Okay, for discovering vaccines using computational methods. Number two is modeling. Actually antibodies, how it looks like a Y shape. So whatever you see this Y shape is a heavy chain, and whatever attached with it is a light chain. But for synthesizing these antibodies, and especially if you see the antibody, the purpose of antibody is in the region, in the antibody region, you can able to see a paratopic region which is going to bind with the antigen. The paratopic region, which is present on the surface of antibody, is going to bind with the antigen, and this is the antibody. So, if you see this kind of a region specifically, we have different SCFE fragments. So, each and every part of this surface is important for antigen binding. So, in two arms of the antibody, may antigen 1 is binding in one arm, and antigen 2 can also bind in another arm. So, other than affinity, which we use it to measure the binding of antigen antibody, we can also measure through avidity. Avidity, I mean, two types of antigens. For example, whatever antibody you use, if it can able to treat, it can able to prevent the measles. Okay, the other and part if can able to prevent rubies, I mean rabies, measles, mumps, rabies, isn't it? Measles, mumps, MMR. We have a multi vaccine called MMR, measles, mumps, and rubella, not rabies, rubella. Okay, rabies we have to use an exclusive vaccine. For rabies for India, Pasteur Institute of India, Punur is manufacturing the vaccine. Okay, they are supplying all over India, and also they are supplying in other parts of the world. If you see this measles, mumps, rubella, a single vaccine can able to prevent three different diseases, and can be the antibody can able to have two different antigen specific binding sites where it can able to the specific antigens like measles and mumps can also bind with it. So one can able to understand that the surface of the antibody is most important, not the entire surface, even a part of a surface. For example, in an antibody, however I drawn, okay, however I've drawn, okay, like a part of a fragment here, this can bind with a specific disease, and here also a part of antibody can bind with another kind of disease antigen. So you can also find such a kind of multiple binding sites on the surface. Okay, where antigen one is recognized in one end and another end. So we can engineer even the surfaces. We engineer the surfaces to get multiple fragments, and each and every fragment can bind with specific antigens. So there is a lot of information needed. You can't able to simply do the modeling. Okay, if you can able to model as normal receptors, okay, but if you want to go with this antibody, we have to go with antibodyomics, especially whatever I mentioned. And other than affinity, where we check the binding of antigen with the antibody. We also have to check the avidity, like binding of one antigen in one arm and binding of another antigen in another arm. Or also, you can engineer the antibodies in such a way that the specific SE fragments can bind with specific antigens. So for this, we need uh, the engineer for doing this kind of engineering. We need this antiproteomics tools. Homology modeling for simply modeling of the receptor, we can use this model. But for antiproteomics, we have to use only commercial tools. We don't have any free tools to do with antiproteomics. The best tool I can suggest is to use Discovery Studio. The Discovery Studio has antiproteomics tool, okay, which can model specifically the antibodies. Of course, antibody is a protein, but antibody structure is slightly distinguished with other protein structures. So to come up with the engineering of antibodies, we need this tool. So if that is the third method, the fourth method is the codon optimization tools. Codons. What are codons? Three letter nucleic acid bases. Okay, that's what we call it as codons. AUG is nothing but start codon. It codes for which amino acid? Methionine. Okay, how to optimize the codon? Okay, so this is actually important based on the uh, coding regions and non-coding regions. You can able to check the translation of proteins. So codon optimization tools are used, okay, to optimize the codon to determine the specific binding sites in the target protein. So if this is step number four, if this is one such a computation method for vaccine design, the fifth method is the scaffolding tools. You can scaffold one biomolecule with another molecule. You can scaffold RNA with a protein. RNA protein complex can be used for treatment of, or it can be used for prevention of specific diseases. Like this, you can use the scaffolds. The biomolecule scaffolds can be used for vaccine preparation. Okay, so these are the various techniques, computational strategies you can use for the development of potent vaccine, including antigen processing. So overall, if you see, the Violin is a web-based vaccine database and analysis done. So whatever I mentioned earlier, VaxQuery, Vaction, VBlast, Protegen, Virmogen, all these are available in vaccine and components. Okay, post responses, we have specific tools. Now, Discord talk. Yesterday I was explaining you the Discord talk we have seen in IEDB. Other than IEDB, we also have exclusive tool, okay, called Discord talk under this URL. So if you go to this Denmark website, okay, specific URL, we have to simply type the PDB code and chain, or else you just upload the PDB structure. If you upload the PDB structure of the specific antigen, for example, Nipah virus W protein, in our case, what we have discussing from the beginning, then obviously we can predict the epitopes, discontinuous epitopes. The next thing is vector builder. The recombinant vaccines for production, especially when I was explaining you something about which is the first vaccine discovered from reverse vaccinology. I told you it is meningococcal polysaccharide vaccine used to treat bacterial meningitis. Okay, so we have taken more than 130 proteins, but during the filtration, like if you want to check how many proteins only can go undergo the recombinant technique, then only few proteins alone are compatible to come up with a recombinant production. So for this, we need the vector. Like we can insert the insert gene in the plasmid to construct a vector. So we need a plasmid sequence and a insert gene, okay, to bring a kind of a vector. So if you see adenovirus vector vaccines for treatment of SARS-CoV-2, how the adenovirus vectors are created. Of course, you are giving a specific target gene. You are inserting a target gene inside that, so that you are bringing a form of a vector, and these vectors are used for prevention of SARS-CoV. So for this, one can use this vector builder. Okay. So revolutioning gene delivery from research to therapy. Next, we can discuss on types of vaccines and diseases treated. If you see, the first thing we'll be discussing is multi-epitope based. What do you mean by multi-epitope based? Linking multiple vaccines. For example, helper T cell vaccine. Helper T cell vaccine linked with the spacer or linkers through another helper to T cell epitope. And this can also be linked with the cytotoxic T lymphocyte epitope. And this can also be linked with an universal epitope. Whatever epitopes we have linked, this is what we call it as multitope. If you find more than one epitope linked with each other, then it's what called multitope. So for the multi-epitope based vaccines, the best pathogen the example they're providing is Kaposi sarcoma associated herpes virus. This is actually linked with Kaposi sarcoma. So how the multi-epitope based vaccine is discovered through immunoinformatics approach. Similarly, for treatment of nosocomial infections through Pseudomonas aeruginosa, so this can able to use comparative proteomics. In Pseudomonas aeruginosa, we have plenty of strains. So if you want to go reduce the nosocomial infections in the lung, obviously we have to go with the comparative proteomic strategy to create a multi-epitope based vaccine. It varies with respect to person to person. The third one is for Ebola virus, DNA vaccine, and for Neisseria gonorrhea, one of sexual transmitted disease. Okay, happened through this kind of a species. This is an in silica hierarchical approach is used. Okay, multi-epitope based peptide vaccine can be used for treatment of gonorrhea and hemorrhagic fever. This has happened through Marburg virus. You know how this vaccine, subunit vaccine. So what is subunit vaccine? A part of a protein which can be used for prevention of specific virus. Subunit is nothing but part of the virus. So it can be a polysaccharide or it can be a peptide, a part of a protein, whatever it can be. Okay, the subunit of a part of a virus. Okay, that's what we call a subunit vaccine. So this was actually discovered through reverse vaccination approach. Human papillomavirus, which is most frequently observed in cervical cancer. 99% of cervical cancer is occurred only to women. 99% of HPV infections can definitely lead to cervical cancer. So for this, structural vaccinology is used to come up with a multi-epitope based vaccine. So like this, we can see few examples for human papillomavirus linked with cervical cancer, cancer antigens generally, especially through dendritic cell vaccines. You know, in my lab we are producing plenty of these. We are doing plenty of research on dendritic cells, especially dendritic cell vaccines. So these are professional antigen presenting cells. If you increase the population in the cell culture, these cell cultures can be infused to the patients to decrease specific type of cancer. Okay, so for this also we need immunoinformatics approach. And similarly, human cervical cancer, this is what linked with viral etiology. Human papillomavirus linked with human cervical cancer. Like this, you can find hepatitis A and B. These are linked with the liver cancer because hepatic is nothing but liver. So for this, you can come up with this kind of DNA-based vaccines, where the strategy used is genetic immune therapy and a pharmacological tool in silico was predicted. And epitope space Zika virus, okay, the corresponding disease is Guerin Barre syndrome in silico predicted immunogenicity. Like this, you can see a few examples. Historic development. So in the latest era, the last 10 years, if you see, plant-based adjuvants are used in existence. Remember I told you. Other than that, even immune proteins produced by the cells can also be used as an adjuvant. Here, the soluble cytokines like IL-2 and IL-4 is mentioned, and similarly granulocyte macrophage colony stimulating factor. This is a growth protein. Especially if you want to grow much of macrophages, you can use this. But IL-4 combination with GM-CSF together, this generate more dendritic cells when compared to macrophages. Actually, monocytes or round cells generally derive from PBMCs, PB peripheral blood mononuclear cells. So, from peripheral blood mononuclear cells to cell culture, on third day, it's going to produce monocytes, which is perfectly round in shape. And these monocytes can further be distinguished in the form of macrophages. If you want to distinguish in the form of a macrophages, then we have to use only GM-CSF in the culture. GM-CSF, granulocyte macrophage colony stimulating factor. But in my case, if I want to produce dendritic cells, okay, if you want to produce dendritic cells, okay, which is having a long arms, this is mature dendritic cells. Then obviously we have to treat the same culture with GM-CSF and IL-4. These cytokines together can differentiate monocytes to mature dendritic cells instead of monocytes to macrophages. Okay, got it. So dendritic cells itself can also be used as an adjuvant. Other than these antibodies, which are responsible for uh, multiplication of antigen presenting cells like macrophages, dendritic cells, including T cells. Adjuvant database, I told you, for coming up with a plant-based adjuvant for a specific disease, we can use this database. And this is what I worked in PhD research. Whatever database I developed, it is working only in intranet. Okay, in internet, we have to post the same. And here in my PhD research, I focused on two important plant compounds from two different plant sources. Okay, whatever plant compounds I use, so this was found to differentiate. This can be used like if these molecules are used, these are differentiating monocytes into immature dendritic cells within short period. Similarly, if a mature dendritic cell has to be converted to mature dendritic cells, the specific antigens taken from BCG, tuberculin, tetanus toxoid separately we have analyzed. So these antigens increase the activation of dendritic cells and even proliferation of T cells. Along with the antigen, if I use an adjuvant, it increases further. I mean, the stimulation and proliferation increases further. Okay, for this, can able to find this. And adjuvants in COVID-19 vaccines. Yesterday, I told you about these vaccines. I believe I mentioned you yesterday. Now, something about functional diversity in the adjuvants. Unmethylated CpG dinucleotide flanked by two 5' purines and two 3' purines. This predominantly has Th1 type cytokine. In fact, similarly, alum, an inorganic salt, can be used for sustained delivery. Okay, if you want to prolong the action of an antigen, we can use this kind of inorganic salt, that is alum. Similarly, you can find this of plant-based AS04. MF59 is submicron oily water emulsion. This can activate the antigen presenting cells. So like this, you can see each and every adjuvant is having a specific action with respect to antigen. Now, if you go with the name, this slide, we see the second wave mutations for spike glycoprotein. I told you we have hundreds of strains of SARS-CoV-2. Each and every mutation can lead to one strain. So here we have first strain, second strain, third strain. You can see the bottom, fourth strain, fifth strain, sixth strains. I have given only the six strains, which is observed more frequently observed in second wave mutations during the pandemic time. Pepfold 37. the most important server to predict the epitopic peptides. I mean, predict the structure of epitopic peptides can be from B cell or T cell. So simply paste the amino acid sequence, and you have to simply click run. That's it. Simple method to construct a peptide. So the peptide in Java editor is shown like that. But if you want to work with any kind of a talking simulation, you have to download from here. Okay, we get two best models, model 1, model 2. Download the model 1 if it is more appropriate, which is binding with the dataset binding site. Now, before going to this slide, am I audible to everyone? Yes or no? Yeah, participants, the last session can interact with me. Am I audible or not?
Yes, sir.
Yes.
Thank you, thank you. Just we have 10 to 15 minutes, not more than that. Okay. Because we are maintaining silence, I don't know whether I have connected to it. Now let me take you to the patch talk. Okay, patch talk. What do you mean by patch talk? This is a protein talking server. So simply you have to give an antigen and antibody as an input. If you provide these as an inputs, we get only the results, don't deepest conformer results of generated. So with the X, Y, Z directions. So whatever we got as results, the top thing is the best conformer, no doubt in it. So whatever output we got like a PDP file, this you have to give it as an input here. Okay, we have to give input here, trans file. And we have to give separate the antibody and antigen separately. Okay, if you give these three files, file one for receptor, ligand two, and trans file is the one the output of the previous patch talk server. If you give this, finally you'll be getting this kind of energies. Okay, for the conformers, the rank one, whatever you find, the specific energies are mentioned. Global energy is this, van der Waals energy of attractive type, and van der Waals energy of repulsive, AC and HB. So these are something about the final results of protein-protein interactions. Okay. Now something about vaccine. If you see the vaccine, I was from beginning I was explaining you something about BCG, bacillus calmette. If it is taken at 8 a.m., that is on early morning, we can find strong non-specific trained immunity. So high cytokines are secreted, especially interferon gamma. You know, the interferon gamma is eminent for treatment of which diseases? If you have more interferon gamma, what you can observe, if you have optimum level of interferon gamma, it can combat what kind of diseases? Anyone know the answer? Interferon gamma is linked with viral disease. You know, you have heard that few persons didn't get any chicken pox, didn't get any kind of SARS-CoV-2 infection also during the pandemic time. How is it possible? How they have maintained such a kind of a stamina? One such most important thing is that in their blood they have optimum number of interferon gamma, which combat all the types of viruses. The next protein which is important, which is secreted in our body through immune cells, is tumor necrosis factor alpha. Tumor necrosis factor alpha. This is eminent to treat cancers. If you want to combat cancer, we should have optimum level of tumor necrosis factor alpha. Okay, so like this, you can see these cytokines are important, and you can see it is highly secreted at on morning at 8 a.m. around 8 a.m. If it is trained at 6 p.m., you find a no trained immunity. Cytokines are secreted in very minimum level, so this is not going to give a good immune response. So if your child is going to take BCG, better we have to take one more. Similarly, influenza taken at 9 to 11 a.m. can yield a higher antibody response. because of metabolic activities and cell differentiation activity and cure to diseases. If you see influenza vaccine taken on evening, you find lower antibody response. What is designed? High cytokine secretion, higher antibody response, but we are getting something negative if you uh, take the vaccine on evening or night. The next thing is SARS-CoV-2. If vaccine if taken at 9 to 11 a.m., we find higher neutralizing antibody levels. Not only that, we find stronger B and helper T cell response. Higher percentage of monocytes and dendritic cells are found. Higher percentage of memory B cells are found. Everything are found to be high here if taken on morning. But if it is taken on evening, we find all the antibody, B cell and T cell response is less. Even percentage of monocytes formed, including dendritic cells, is less. Okay, all the immune cells are producing very less if taken on evening. I believe the Covishield and Covaxin, whatever you have taken, is on morning. If you have taken in evening, obviously the efficacy is going to reduce. The next thing we'll be discussing is antibody modeling. I told you for antibody modeling, you can't use Swiss modeler, ITSR, etc. For antibody modeling, the only solution is to use the commercial tools. In the steroid injection, we have a specific commercial tool for antibody modeling. Similarly, for DNA star. So both are commercial softwares. Okay, but these are unique in developing in modeling the structure for antibody. So that's it. I have completed my session today. Now let me take you to the websites. Okay, and I give you a few illustrations. Do you have any? If you have any doubts, you can ask me. In theory, don't have any doubts. So let me take you to the two vaccines. So if you see the human vaccines, we have to select specific thing. Okay, like I'm going to take chikungunya virus in humans. You know that for 5 years before for chikungunya we can able to find the vaccine. So at present, for chikungunya virus we have the vaccine for prevention. So simply I selected one specific virus out of numerous viruses. Click search. Okay, it asks you like license chikungunya human vaccine. The vaccine license vaccine ID is also mentioned. If you want to go further, select this compare. So if you get 2 3 vaccines, you can also compare all these 2 3 vaccines and you can choose which can be used in your location. So this is licensed chikungunya human vaccine. Similarly, we can go with other tools. Like our search for human vaccines, we can search for veterinary vax. So if I click VVAX, it is taken to this kind of a link. Simply select a specific animal. For example, if I'm focusing on chicken, okay, and I want to check avian paramoxy virus, okay, so I'm selecting these two fields. Search. We got like for the selected two fields, we got three types of vaccines. Okay, pathogen name avian paramoxyvirus. We have three vaccine steps, so you can get the information related to this. Okay, by getting inside. Okay, this is the vaccine ontology. Okay, like this we have few more tools. Protective antigens, protein check. We have to select specifically one particular pathogen. Now I have selected Bacillus anthracis. You know this Bacillus anthracis infection got spread in IIT Madras frequently. During the pandemic time, we can able to see that kind of a spread. And even I believe 2 years before, the same kind of infection happened in IIT Madras campus. Okay, so how the anthrax is formed? Only through the spores released by Bacillus anthracis. So now I select Bacillus anthracis. and we can go with the specific that COG category. If you are going with specific post-translational modification protein turnover, select that COG. Cluster of orthogonal groups. The expansion of COG is cluster of orthologous groups. So we have to select a specific group there. If you want to specifically target a membrane, okay, you can give a membrane or other parts and simply have to click search. This is used to determine the productive antigens. For the specific thing, we don't find any productive antigens. If data is available, it shows, or else you'll be getting the image like this. I mean, you will be getting the output like this. Similarly, if you see, next thing I'll be taking you to the PBI lab, Protein Bioinformatics lab. Okay, this is maintained by my mentor, postdoctoral associate mentor, Professor Michael Bromina. He is also one of the popular researcher, professor in top university in Japan. Okay, so if you see his kind of developments, he has developed so many kind of tools and databases. He and his PhD scholars has come up with the tools and databases for almost all the biomolecules. For example, the top link what is should see is the DRLIPS. This was developed by these kind of research scholars on the guidance with the guidance of Professor Michael Rubin. If you see this tool is used as a reliable method for predicting binding affinity of RNA small molecule interactions using mission mode. If you want to check binding affinity of RNA and small molecules, you can use this tool. Similarly, for mutation prediction and zinc finger proteins in cancers. Okay, and then we have specifically reliable method for predicting RNA molecule interactions, as you mentioned earlier. So, like this, we have plenty of tools developed for interaction of lipid DNA, lipid RNA interactions, lipid protein. So, like this, we have different kind of biomolecular interactions developed by him and the tools. We also have specific databases developed by the same PhD scholars. For example, ABCOB. This is a curated database for binding affinity and neutralization profiles of coronavirus related antibodies. For this, you can use ABCO. Similarly, we can able to find plenty of tools available. Okay, these tools can guide you like how to come up with epitope-based vaccine design, or even the interaction of DNA protein or RNA protein or lipid protein. So all these interactions are perfectly that data you can perfectly can able to see in these databases. The most important is go to the protein platform fix lab, go to the resources. And what else did we see here? Bioinf. This hyperlink can tell you the different tools used for vaccine design. It's getting loaded. Meanwhile, I can close the windows. I think the server is down, so it is loading slowly. So like this, we find plenty of tools here. You can check the tools and simply select queries. Okay, and search. You will be getting the information. So today I have completed the session 10 minutes earlier. Okay, on co- tools and servers for computational vaccine design. For case study, we already discussed with respect to Nipah virus. So how to predict T-cell epitope-based vaccine. Okay, we already discussed. So now there is no need to discuss. If you have any doubts in today's session or previous session, you can ask me this last 2 minutes. So one thing you have to remember that 25 MCQ questions and feedback will be circulated on today through mail and WhatsApp. So you can try to complete it on or before Saturday. I mean that you will be getting the certificates on or before coming Sunday. Okay, that is on. You'll be receiving the certificates on or before 2nd of August. Okay, based on your completion when you complete both the forms. Okay, any doubts? You can ask me. If you don't have any doubts, okay,
No, sir.
yeah, you can ask. No doubts. Okay, that's what he has mentioned. So if you don't have any doubts, okay. So thank you very much for your cooperation from the beginning. You maintained silence and also you interacted whenever I asked the questions. Okay, so we are coming to the end of this certificate course. We need to yes, it's needed. Both we have to submit both the quiz and the overall feedback. Okay, so we submit both. You'll be receiving the certificates. Okay, thank you. Have a nice day. Good night to everyone.
Thank you, sir.